Very happy and honored to have Professor Peter Falkai with us today. He worked with us this morning to try to make and create a consortium between expert centers, French-mind centers in France, and the German centers of excellence. And I don't think he needed any introduction, but just in case some of you don't know him, he's Professor of Psychiatry at the University of Munich, and also affiliated to the Max Planck Institute, and he's coordinating this network of German centers of excellence that he's going to describe. And we've asked him to explain how science and knowledge is translated into clinic, which is a big question for people like us producing good science. How do you clinicians, how do we clinicians use it? Peter, thank you very much to have made the trip to here and to bring this. Thank you.
Marion, first of all, thank you very much for the invitation, dear colleagues. Being I call this clinician scientist, I personally like to speak about translation. And when preparing for this talk, I thought I give you two examples. Well, one where it didn't work out well, and the other one where it sort of partially worked well, just to think about, you know, what are possible things we need to tackle if we want to be successful translators. So what is translational research? I will start with that. Where does translational research stand in Europe? I always like to look at that. I have some interesting numbers. France is in a good position, just to start with. The translational gaps, ways ahead, and I think translation needs role models. Translational research has been characterized as harnessing the use of discoveries from basic science to develop new diagnostic tests, therapies, and prevention devices, sometimes referred as type 1 translation, as well as the implementation of research findings into practice to improve care for patients, type 2. So it's not enough to actually have a discovery and then, you know, have new diagnostic tests and therapies. We know that there are a lot of things around, but they never reach the clinic whatsoever. And but as a clinician scientist, I would say to bring something into the clinical routine is, I would say, and I'm now almost 35, more than 35 years in the clinical case, I haven't seen many times, to be honest, in psychiatry. Now, where do we stand? We actually look worldwide, research-wise, and you immediately look at US because you think they're doing a good job. Well, they're doing a good job, but I think that Europe overall does not a bad job. And then you look down to United States and so forth, then you can see we're not that bad in Europe. And if we tackle down the countries, I wanted to make two things, but you know, Germany and France is close, and France is even significantly better than Germany. So I always say, if you look at, that's the number of studies done. The question is that a good marker actually to use that and say, oh, we do many studies, that's why we do translational research successfully? Well, you can use that if you want to convince people to give you more money, but the question is sometimes you need to think about, okay, what are we doing there? And you know that we have these valleys of death of psychiatric translation. One is a type 1, and the other one is the type 2. And there is a paper, well, you can't say recent, but way back in 2008, when they actually looked at research and developed expenditures on one hand, and on the other hand, new molecules. entering the clinic, and you can see there is a certain divergence. Or if you actually look for people being interested in translational research, that's even another gap there. But I'm now telling you a story where we hoped that we will be successful, because, I mean, if we speak about precision psychiatry, what do we need? Two things: mechanistic understanding of the illnesses, and the second is having biomarkers in order to subdivide our heterogeneous group of patients, to make sure that we have subgroups whom we can then target, let's say, with a specific treatment. Well, we have started another way. We started at basic science and thought, you know, what are obviously deficits in schizophrenia? And there is the so-called disconnection hypothesis, and sort of the hypothesis that there is a cortical disinhibition, or if you want to say so, a disbalance between excitatory and inhibitory elements. A lot of people would agree on that and say, well, that sounds nice, but the question is, how are we going to research that? Well, you can look at certain interneurons, like the ERBB4 interneurons. And so the hypothesis was, if these are less excitable, then we have increased activity of the cortical projection neurons, and that causes problems. Well, we like easy to explain hypothesis because you can prove or disapproves. That actually leads to a disbalance of excitation and inhibition. We go into the whole system. You can see indeed, as a consequence, we would have an increased dopamine release, positive symptoms, or a decrease of dopamine release in the prefrontal cortex, causing negative symptoms and cognitive dysfunction. So the goal was here clearly set. What we want is actually enhance activity of these interneurons in order to restore excitatory inhibitory balance. Sounds good, doesn't it? At least for a grant, it's good. So what are we going to do there? So these are these ERBB4 interneurons, and you can actually view them. They have their special position, and if you actually look at the glutamatergic neuron, then there is the inhibitory interneuron. You know, this is how they place themselves, and that goes and modifies glutamatergic transmission. So this is something, if it works well, this is fine. We need glutamate all over the brain, and you know, if we function well cognitively, emotionally, and so forth, glutamate is important. Okay, but how can we do that? And we actually targeted this so-called NRG1 ERBB4 pathway in order to re-establish the imbalance. So what might be sort of a possibility to do so? So if we have an imbalance, and we actually hypothesize that that is true as an underlying cause if people are psychotic, or let's say have cognitive dysfunction, and we decided not to go for psychosis, but for cognitive dysfunction. So the idea was, because we have a stable imbalance between excitatory, inhibitory, let's say, mechanisms, people will not be able to actually have a good cognitive functioning. And that's another step you can go. We then did drug repurposing screen, and as you can see, we've screened a few thousand drugs which actually are applied in humans, and found 2-3 of them which actually modify exactly sort of this ERBB4 pathway. Good. Spironolactone, we know it's for peeing. So we thought, okay, why not giving it a try? So the try was then to validate that in a mouse model, right? So we have looked at that, and indeed we could show, and you have to believe me, that adding spironolactone normalizes actually inhibition and disinhibition and excitation. So from there, that was a cellular model. And then we went into the mice. So starting with the mechanism, then going into a repurposing screen just to modify this mechanism, then go into the cell. So if it wouldn't have survived the cell, then we would have stopped, but it survived it. And so we looked for the NRG1, neuregulating 1 transgenic mice, and added spironolactone to these guys. And you can see overall, there was an improvement with increasing dosage. So it was interesting that indeed there was now ample, let's say, evidence that that should work. So we were very proud and thought, good. So now we have the mechanism that has been proven in cells, in animals, and now we go for the clinical study. And it was one of those things where I bet this is going to be the drug against cognitive dysfunction in schizophrenia. So we did a 3-arm study, and maybe that was the beginning of the sort of death. And we have actually sort of introduced spironolactone, and as I pointed out, which helps you validate it in the other principles. Symptoms to target, while positive and depressive symptoms are viewed to be effectively treatable with available antipsychotics, the efficacy of such compounds in the measure of cognitive symptoms is not persuasive, to say the least. So what did we do? That was the so-called Spiro trial. And you can see up there, so you have the usual sort of washing in phase. You had a 3-week period where you gave 100 and 200 milligrams. The fear that people would pee a bit more didn't happen. That was quite interesting. And the internists who helped us in the 3rd arm was placebo. And the hypothesis was that clearly, especially the 200 milligram dose would improve cognition in schizophrenia. And then we had a stable phase, and you could see the pre-screening and all the things you need to do there. And so overall, the idea was, okay, let's push for that. These were the sort of things we did with the patients. You won't be so interested. Our primary outcome, and maybe this was a bit courageous in addition, was working memory change. But I mean, if you want to research cognition, you go for working memory change, right? Inclusion criteria as defined, so the usual suspects, exclusion criteria. So there's nothing really to mention about. So a straightforward clinical study. Never do a 3-arm study. But anyway, at that stage, we thought, go for it. So just to show you the timeline and the distribution, we had a 3-center study. Always include centers where you are sure that they include patients. You see, this was our center with 75 and 2 centers with 12 and 5. Anyway, that's life. So then you look at the psychopathology. Then you had, certainly, let's say, mildly sick patients, but we wanted to have them clearly scoring on the negative symptom score, which they did. Safety. So we have looked at patients with adverse events or change in drug treatment, and overall there was no big thing. So you would say spironolactone is well tolerated by the patients, which is good news, because sometimes you think, well, you never know, and add in that. And if you look at the data, you would think, okay, that looks nice because we have stairs, right? So if depending on the amount of spironolactone you give, you have an improvement of the NBAC, sort of the working memory paradigm, in a stepwise fashion. Because as you can see down there, so this is V1 to V12, and overall you can see that actually gives you the impression, and if you look at that, you would say, well, this is nice. Unblinding the study, it was completely negative. So we started with a hypothesis and, used repurposing and did everything just to make sure that we're landing in the clinic. So what did we do wrong? Nothing. But this is the kind of studies you need to do. And also, I'm not really a fan of Insel, but I like what he says, and I made rethinks psychiatric trials, genetic pathways, relevant model systems, subphenotyping and genotyping. That is clinical studies based on biological mechanisms. I will allude to that a bit later. So, okay, so we died in the first valley, right? But that can happen. But I would, and I'm at the moment working on another trial. We started a trial where we look whether clemastine, what is this? Clemastine is actually a drug you use, it's an anti-allergic. And we use it in schizophrenia as an add-on. Why on earth do we do this? We do this because we have seen that actually myelin plasticity is disturbed in schizophrenia. And we have that replicated in several studies using exercise. And we could show that exercise actually improved myelination to the extent that it normalizes your sort of connectivity networks, especially the salience network and other networks, which are important for functioning, for symptoms, and so forth, in about 50% of the patients. And logically, if you have a myelin problem, you go to MS, right? And if you go to MS, you can easily show that clemastine gives an effect in a 90 days trial. There are 2 subsequent trials. So this is when I thought, let's use clemastine. And the interesting thing there is that we even got money, because that's the other thing, is you go to, a foundation and they say, clemastine, psychosis, are you crazy? They don't fund it. And I found a foundation who said, quite crazy, but good idea, go for it. So we are now doing this study, and the question is, I can't tell you any results, that's why I didn't present it, it's just I wanted to give you the feeling, certainly I'm not giving up on this, because I do believe if you go for mechanistically based treatments, you will understand the illness and certainly pathways into the illness. What about the second, the type 2, let's say, translation? And I think we are better there, at least I give an example. RTMS and psychosis. If you actually look at that in the literature, there is meta-analysis clearly showing that RTMS helps in negative symptoms. Okay, we thought this is good. So we asked for money and got approval and did a large-scale study. So what did we do? Screening in, 2 weeks assessment, and then 3 weeks, 15 times RTMS. And the hypothesis was, we target negative symptoms and we improve this. Based on the meta-analysis, this was a clear game, right? And you can see we screened almost 200 patients, randomized 175 in order to end at the end of 73 per arm. So what was the design? What was the result? Completely negative. Show that on purpose, because it will have a good ending. So, you look at this, and both arms are getting better, the sham and the RTMS. So it's good that you have a control, that you have a large controlled study. That's the largest in this field. And we stopped at least using RTMS in negative symptoms. Well, we've looked at positive symptoms, total symptoms. We looked at depression with different one and functioning. No difference at all. Okay. So you could say, done, shot, right? But something which sort of convinced me there's something in it is these big bars. So there is a lot of noise in there. So obviously there are some guys who do respond. And isn't that what we want for the clinic? So we had actually scans of the patients. And this is work of Nikos Koutsouliris. So they said, can we actually predict response? And if we predict response, which of the brain regions might be involved there? Precision Psychiatry. And it was interesting that those guys who got better actually showed volume changes in the hippocampus and in the parahippocampus and other relevant cortical areas. So it was interesting to see that if it works, obviously the networks relevant for psychosis are activated. But what can you do? How can you decompose the outcome heterogeneity? And we used machine learning applied at the baseline of the MRI. And what was the result of that is that. So you could actually disentangle with using the MRI those guys in prospectively who would then sort of respond. And you can see here you see the responders quite nicely, and here you see the non-responders. And it's about half of the patients. So I have to say that based on that, we have done a prospective study and could replicate that. That was just published recently. The only problem with that study is you needed an MRI scan. And, you know, doing clinical work just to do an MRI scan to decide whether you do RTMS and so that won't work, right? But it shows in principle that you can use data in order to predict outcome. And that's another sort of facet. We have actually looked at the question whether you can detect with clinical data the occurrence of depression in a GP population. Well, you know, GPs, at least in Germany, they actually treat a lot of people with mental disorders. And you can say roughly 10% of these guys have depression. But in most cases, this is never recognized. They just have it, and then they're treated for high blood pressure, they're treated for gastrointestinal problems, they drink sometimes and things like that. But no one says they have depression. And we used clinical data, and with a likeliness of 93%, we could actually identify those guys who had depression then on the scales. So my message would be, yes, you can look at, let's say, predicting diagnosis, and you can go for outcome by using the data you have. And I think we should possibly not start with genetic, with imaging, and so forth data to do prediction, but maybe use everything that includes clinical data. In this case, that didn't help. That was only the, let's say, that was the scan which actually helps. So the question is, you know, what factors enable and hindering translational research? Well, I think I like that paper, therefore I took it with me also. It's older. So if we actually look at the concept of translational research, often I think it's overburdened. That's why I brought the first study. You know, we had this hope, we have the mechanisms, we have the animals and everything. So that needs to work in humans. Sometimes it doesn't, right? The research process, you know, how we actually recruit patients, whether that works out nicely. Then sort of the clinical care. Can we recruit enough patients? And then sort of what role actually translational research has in a surrounding. So I think that translational research needs to be circular and an iterative process. I mean, I've pushed my people to actually go for the first study and actually lose another comparator and go for a two-arm study. The problem is, if I'm not writing up the grant, no one will do so. So I think not giving up on a thought is one thing, and just to continue and actually have a feedback on that. I think organizational and institutional level, actually streamlining regulatory governance processes, governance, we spoke for lunchtime, is one of these big things. What about the infrastructure? Is the bioinformatics sufficient? Otherwise, you know, you have nice data, no one actually does the processing. And then academic and hospital institutions. I mean, I have the privilege in being in two, which actually are split, right? But my impression is sometimes I do a ward round and there's very few people who are interested in research, and most of them, well, they think, let's speak the old, or let's listen to the old man. Maybe there's something interesting in what he says. And I think that's another thing. How can we attract people that research is something nice and something which can sort of inform your clinical work and vice versa. Stronger leadership and integrated structure institutions. Well, stronger leadership is something which then possibly I should look at. How can we solve that? I mean, just you know, complaining about things I think is absolutely not useful, and it certainly is not my style. I think that there are a few things to solve that. One is use computational sort of psychiatry, using large scale data, and actually then bridge the, let's say, at least the hypothesis to biology, as pointed out here, and simplify. That's the other thing is, I think that often clinical models are much too complicated. And in a way, phenotyping modeling of the neural, let's say, robotic platform. So to make sure that we do understand what's going on in the brain. And I've just brought a very simple example. Well, you could say epilepsy is not psychosis and not depression, but I always like to say, well, at least it's the brain, right? And so they actually used data in order to model the process of epilepsy and simulated the epileptic EEG signal compared to normal EEG signals. But that was based on data they used and actually built up models on that. Well, what about if we go back to psychoses? What about building up models of understanding schizophrenia, well, based on the old dopamine hypothesis of schizophrenia? You can actually go through and build different models. I'm often thinking that computational neuroscience and computational psychiatry often is not really helpful in the clinical context because we don't understand them or they don't understand us. So for me, it's always important to actually raise the questions from the clinical side and then understand what these models mean and where their actually borders are. Neuroimaging genomics. I think that is by all means a very interesting way because it's clearly sort of looking at the sort of risk we're looking at, because I think that mental disorders are basically genetically driven brain disorders. There is a certain and a significant amount of environmental risk, but I mean, no question, there is genetic risk. Now, the question is, what is always put up, what about our diagnosis? Is that something we should look into? So beside using things like computational psychiatry, what about other systems? When I've read RDoC for the first time, I thought, I mean, do we really need this? And what is the underlying neurobiology? But the more I actually get into it, I think it's a way of disentangling. Possibly it's not that easy, as outlined, going from negative volumes, positive volumes, cognitive, social processes, arousal, and then actually have the unit of analysis, but it certainly is bridging different diagnoses. And I personally think that, you know, coming from the German Center of Mental Health, we will use parts of HiTOP and we will use parts of RDoC. Reason for that is that we like to look at healthy people at the early phases of mental illness in an early phase and later phase. And it's very difficult to have scales which actually mirror that. That's one thing. The other thing is we'd like to include many patients. And as you can see, if you have a lot of scales, it takes a long time and you will have a high dropout rate. At the moment, we are, for each patient, self-rating 20 minutes. So HiTOP is a different way of looking at I mean, that was actually launched by psychologists. And as you can see, it's difficult to see there are different psychopathological domains, and I personally think that bringing this together, a hierarchical, hypothesis-free way like RDoC, and then another one where you with high top stick to the psychopathology might be a way, let's say, ahead. And the question is, has that shown that it works? Well, I personally think that indeed, if we look at papers coming out of, let's say, the RDoC system, just looking at the results, that they looked at different genes, they looked at molecules, they looked at circuits and physiology, they actually nicely bridged, let's say, using fMRI, using these paradigms. Again, RDoC, if we look at that and we compare that with the, let's say, diagnostic systems we use, actually, I think there is convergence if we look at categories, if we look at the concepts. So overall, I think we should make clear that actually DSM and ICD, we need them for diagnoses, certainly to be paid. But I think if we do research, I think we should think about RDoC and HiTop. Big data. I mean, that's why I was here today in the morning. So it's not only enough to have, let's say, the data of a country like France, which is big enough, but we want to come together and harmonize and actually use the data of the German system, of the French network, let's say German network, French network, and possibly even joining the English one. And I personally think that that actually helps to then look for generalizability of the results. And as a clinician, and then I just give a little outline what we do in the German Center and then stop. I personally find it interesting that if you actually look at large scale, let's say, data, you come up with a 4-factor model instead of a 3 or 2-factor model, clearly showing those domains which are important in psychosis: positive, negative, cognitive, and affective. So, role model is maybe an important thing. I think if we speak about translation and translational research, I think, at least in Germany for a long time, we didn't have a role model for that. We had little studies, we had some centers, you could go there, but we had 10 years ago, the so-called health centers established. And I think that that has actually helped a lot to bring health into the focus of research, especially translational health. And you can see the already existing centers 10 years ago: diabetes, cardiovascular, infections, pulmonary, neurodegenerative oncology. And when I gave talks at the end, I would always show that in German, that is beautiful and it's nice, but we have only one problem. There's mental health lacking. Well, it took a few years, but then in 2019 or 2018, they thought we have 2 more health centers, one for child health and the other one is for mental health. So what did we do? Actually, you had to apply. It was a several stage process, 3 stages. First stage, you applied as a local consortium. This consortium actually consists of LMU, TUM University, Augsburg, and then you had Max Planck and Helmholtz. Sort of everything in, let's say, mental health in this region. We were successful and became one of the 6 partners of the DZPG, which encompasses Bochum, and then you have Halle Magdeburg, you have Mannheim, you have Tübingen, you have Munich. So these are the 6 centers, which were actually then finally selected by an international review. Do we have more centers of excellence? Well, I think so, but that was the way these centers were, let's say, selected. Is that the final setup for until the end of our days? No. There is, after 10 years, a quite thorough sort of review when there are the chances, if you don't do your job well, you actually have to leave the round and other centers will enter. We will have more centers? Well, the idea is that you actually can associate other centers with these 6 centers. For us, important is participation and co-creation. That, in a way, was different with the other health centers. As you know, I mean, we have, if you want to say so, in the beginning, had to learn how they do their own. So I just had a phone call in between because we have a board meeting, which is running fine. And one of the questions is, how much participation do we have? Should patients and their relatives have the right to have their own studies without, let's say, professional PIs? And well, yeah, I mean, depends on, you know, how you look at it. I personally think it's a team effort. So I don't think that any study should be done excluding anyone. I think in every study we should have early career scientists, we should have a PI, and we should have patients and relatives. That is at least my opinion. Okay, we'll see how we come there. But it's interesting aspect. Looking at the research program, we have, let's say, put it around 3 topics. We said risk and resilience factors. The second is innovative individualized treatments. And the third is actually putting these treatments into everyday clinical use by prevention, early prevention, looking at recovery and participation in living environments. So that's in a way different from your center where you foster research. This is more like a top-down approach where each of the centers gives their program, but we have an overarching program. If you actually look at the first domain, which is risk and resilience, you can see we look at things like trauma, we look at things like lifespan psychosocial trajectories. If we look at interventions, we look at digital interventions, personalized pharmaco and psychotherapy, and targeted stepped care. And living environments, really, we want to, and we started to do population-based studies and actually look for recovery and related mechanisms. We have then said, okay, we get a lot of money. This is a program. Is that program ambitious enough? What are our really goals? What are our flagships? Is like, you know, going to the moon or something which is not within the range. And so we had 3 flagship projects. One is urban mental health, because we think that understanding urban health, or let's say the urban surrounding, then we understand why people in an urban surrounding have a higher likeliness to develop mental health. Sorry for that, but I'm coming from an urban surrounding too. The second is early recognition and intervention and prevention. I think that was an important topic. We are building on a, let's say, network there. And enhanced psychotherapy, we called it, if you combine psychotherapy targeted, for instance, with esketamine and other things, you know, are they better, are they different? Which kind of patients do they meet? And then we call the thing at the end, one mental health platform. Well, the dream would be, if we look at that from the meta level, where we look at the human-animal interactions, interventions, climate change, and biodiversity, just having mental health as part of that thing. So that was the idea where we said, you know, we need to pick up certain things. My group has sort of engaged in the Flagship 2 project, but I think that that is quite evenly distributed among the 6 centers. We have actually included like people like you quite at the upfront. So we got some more money from the for the next 5 years every year, and we use this money that the early career scientists can actually use that money and have their own projects. They should include PIs, they should include patients and relatives, but they are the principal investigators. So they do the study, they design the study, they're responsible for the output, they do write the paper, but they get support. And I thought that had some lengthy discussion. Is that a good way to go? I think it is a good way to go. What is important is linking up with other centers, at least locally. For us, it was important to link up with the, let's say, the child and youth center. Why? Because I think there is a clear road from, let's say, childhood to later adulthood. And we need to recognize that 75% of mental illness problems start between 16 and 24. So I think it would be a mistake if we leave out, let's say, the early antecedents of this illness. So I'm almost done. I think that if I think about translation, I would say cooperation between university and non-university care should be improved. I think that we should make clear what are our goals, what is really innovative. I've given you 2 examples where I think that, you know, to work on that and continue. And I think that I'm always liking to look at structures which obviously are at least better than what we have in Germany. I like to look at Karolinska, because Karolinska in itself is an interesting center where they sort of bring together research. Look at KCL and so forth. But I think there is, on the other hand, another side to that. I think having a center like you have, where you allow diversity and different directions, and I think that has its beauty too. On the other hand, the output of these huge centers, thinking at the Karolinska or KCL, well, at least in Germany, we can never reach that. So this is my conclusion. If we look at barriers, a lack of culture of translation within institution, inadequate infrastructure, and inadequately trained workforce are, I think, the barriers. What are the requirements? I think collaborating with other institutions, well, a culture which actually not only divides, but brings together scientists and clinicians. I think the term clinician scientist nicely brings that together. And the other thing is the reward. I sometimes feel as a clinician scientist, there's not a lot of reward to doing this. The basic scientists say, are you crazy doing research with humans? They're much too difficult to do research on. And then let's say the clinical researchers say, okay, you should look for drugs, you should look for things, low-hanging fruits, and then trying to do and actually reaching, or let's say going over the gaps I pointed out. I think that's a lot of fun, but on the other hand, I think you need a lot of energy and you need to believe in it. I think that's most important, that is the way to go. If you don't believe it, you won't do it. I personally do believe that bringing actually mechanistic understanding in mental health and actually introducing mechanistically based treatments, that's the way ahead. And why? If you compare that, for instance, with oncology, cardiovascular medicine, and so forth, look at oncology. I mean, where have they have been 15, 20 years ago? And how did they improve? And I think they improved because they understand more of their illnesses and they actually use mechanistically informed treatments. With that, I'd like to conclude with an old picture. This is Alois Alzheimer, and he is surrounded by scientists. And what I always like to point out that people like the Louis Body-Louis, he worked there, and other scientists. And I think obviously, even at that stage, which is more than 100 years ago, they do believe that what they work on is the brain. Thank you very much. So the question is, spironolactone is an antagonist at the mineralocorticoid receptor. How do you explain that activation of those receptors with fluorohydroxycortisone was documented to improve also cognitive function in various pathological conditions. In addition, did you explore the relation with cortisol profile in schizophrenic patients? Okay, well, I think that's a good question. We have actually looked at that and found no correlation overall. And the first question was about the how do we explain that the activation of those receptors was documented to improve also cognitive function? Well, I mean, there is, I've just shown some evidence, but I think there's evidence from other animal studies and there's evidence from human studies that actually that substance seems to improve cognition. So for us, it was a good, let's say, candidate to look at. But I think if you ask yourself why we failed, I think, you know, we were too brave, 3-arm study. And the second thing is possibly, you know, we need a larger power, a larger N. But I say, if you do a small scale study and don't find a difference, I think you might think about stopping that avenue. And but on the other hand, I think, you know, I do think that we need to start from mechanisms and go into clinical studies. And even if we fail, well, that's it.
Just a question regarding your last remark. You say that you can try a new drug on a small sample of patients. If it doesn't work, it's better to stop, continue the process. And does it fix? My feeling is that it fits against the precision psychiatry, because you suppose that maybe one drug could be interesting for a subgroup of patients, which would be highly difficult to select, maybe. And how do you reconcile those views? First starting with a small group and then trying with a very large group.
Well, my answer is on the second study I showed you. When we looked at the standard deviations, and that was huge. When I saw that, I said there is music in there. So if you see something like that, a high standard deviation, I would go and look into, let's say, the distribution of the patients. I mean, even in the small study, I would always look at the distribution. You know, are there some guys who do respond? My only problem is, you see, you're going from a small study, which actually does not give you any signal, to a large scale study. The question is, who's going to pay for that? No one. So I personally think that, you know, if you're very convinced that that's the way to go, you should continue. But I have to say that also I was convinced about the mechanism. I'm still convinced. But these clinical data, well, at least for us, I mean, what we should do, we would use another comparator and would go for a 2-arm study. But you need to actually have someone who has the courage and the strength and the power to actually apply for money with this negative study and then say, okay, I have good proof that it works. That's the problem. And the other thing is, you see, on the other hand, sometimes you need to actually accept that it doesn't work. I mean, I'm in this field for more than 35 years. I have had many, many negative studies. You know, when I turned 60, I invited good friends and former pupils and things, and they gave talks. And one of those guys sitting next to me said, but these are studies, most of them are negative. And I said, yes, it is. And this is where we stopped then research and swapped. But I think that I never stopped being a, let's say, clinician scientist, because I do believe that we need to improve diagnosis and treatment. And I think that small scale, well done small scale studies are the way to go. Because if you have a signal, then you have the power and the arguments to go the next step. Then you go for maybe 2 or 3 centers, and then you go into implementation.